Good morning, everybody. Thanks, Lara, for your kind invitation. Really, I'm very happy to be part of this inaugural symposium, even remotely. And as I said, I listened to most of the talks, and they are all really fascinating. So today, I'm really happy to share with you the work we have done on Galectin-Glycan interactions at the cell surfaces. And most of you know that galectins are glycom decoders. They are involved in many biological functions, such as development, tissue regeneration, apoptosis. They regulate immune responses and inflammation. And on the pathological side, they are also involved in cancer development, because many tumors overexpress galectins, so they play important role in tumor progression and metastasis. And galectins are also able to recognize glycans found on microbes, so they are also involved during infection. So what defines a galectin? So all the members of the galectin family bind specifically to beta-galactosides, and they all share a common carbohydrate recognition domain fold. And this fold is the following. It's a beta sandwich with describing two sides, one convex and one concave side. And within this side, you can the glycan can interact, and the carbohydrate binding site can be divided into 5 subsites from A to E, with subsite C and D contacting directly the galactose moiety, and the other subsites are basically interacting with the extension of this galactose. So galectins can be classified into 3 subtypes. The prototype, which consists of 2 CRDs, homodimers, the chimera type containing 1 CRD and an N-terminal tail, which can pentamerize, and the tandem repeat galectins containing 2 CRDs, 2 non-homologous CRDs linked by a polypeptide linker. So as you can see, galectins are multivalent, and this multivalency helps galectins to achieve their functional interaction at the cell surface. They are therefore able to mediate cell-cell interactions, also cell matrix interactions. They can cross-link receptors at the cell surface, and they form therefore lattices that can trigger cell signaling inside the cell. So galectin-glycan interaction have been extensively studied, and actually we have hundreds of structure between different type of galectins in complex with different glycans. But however, given the abundance of beta-galactoside derived glycoconjugate found on the surface of the cells, how this lectin can select a subset of ligands among many available candidates remain an open question. And this is a key question, because this selection of a specific glycan at a cell surface is going to trigger a specific cellular response. During the past years, there was several reports, and actually the concept of galectins as functioning in the extracellular compartment only through carbohydrate interaction has been challenged by the identification of non-glycosylated binding partners. And whether this interaction can interplay with the glycom decoding function of galectins is not known. So in order to decipher this interaction and to understand the role of this interaction, we did some work. We studied the galectin-1 interaction to a pre-B cell receptor during early B cell development. So very briefly, early B cell development takes place in the bone marrow, starting from an hematopoietic stem cell which is going to differentiate into a pro-B, a pre-B1, pre-B2, and immature B cell. And the first checkpoint of this differentiation is at the pre-B2 cell stage, where pre-B2 cell can interact with stromal cells secreting Galectin-1. At the surface of these pre-B2 cells, there is a pre-B cell receptor that is expressed. And this pre-B cell receptor needs to be clustered in order to be activated and to induce signaling, which will lead to the next step of the differentiation. And this clustering can occur at the contact zone between the pre-B cell and the stromal cell here. And it has been shown that Galectin-1 secreted by the stromal cell can interact with glycosylated receptors found on the pre-B cell and on stromal cell. But also, Galectin-1 can interact with this N-terminal part of the lambda-5 protein from the pre-B cell receptor. And this interaction is carbohydrate independent, meaning it's a direct protein-protein interaction. So all these findings led to the pre-B synapse model, where Galectin-1 binds to glycosylated receptors on pre-B and stromal cells, but also interact with the pre-B cell receptor, and this network of interaction drives the pre-BCR at the contact zone between the two cells to induce cell signaling. So, of course, as a structural biologist, we wanted to know what the structural basis of this interaction between Galectin-1 and the pre-BCR. So how a galectin can interact with a protein, a non-glycosylated protein. So to answer this question, we use NMR spectroscopy in order to solve the structure of the complex between Galectin-1 and this N-terminal part of the lambda-5 protein from the pre-BCR. And here is the structure that we got. So if we look now at the structure a bit more closely, we can see that the peptide is binding at the edge of the beta sheet, the convex side of Galectin-1, and at distance from the carbohydrate binding site. The residues involved are mainly hydrophobic, with this tryptophan completely buried into the galectin structure. So you see here the peptide and here the carbohydrate binding site. So there are the two binding sites are really distant. So now, having defined the structural basis of this interaction, identifying these hydrophobic residues, our collaborator, Claudine Schiff, tested this hydrophobic contact in a cellular assay in order to check the pre-BCR, the impact on the pre-BCR relocalization. And she could clearly see that mutating this tryptophan 30 really impair pre-BCR relocalization. So this hydrophobic interface is really crucial for Galectin-1 dependent pre-BCR activation. So the next step was to answer the question about the impact of this peptide binding on Galectin-1 carbohydrate binding activity. So in order to answer this question, we use a Glycan array screening for Galectin-1 in absence and in the presence of the lambda-5 unique region. So as you can see here, we compare the profile, and as you can see here, we see clear increased binding for several glycans, whereas others show decreased interaction with Galectin-1 in the presence of the peptide. And in order to validate these changes, we took two of these glycans and tried to quantify the affinity changes. So this work has been done by Jeremy Bonzi in my group, a PhD student. So we tested using ITC the affinity of Galectin-1 for this tetrasaccharide, the LNNT, which shows a decreased interaction to Galectin-1 in the presence of the peptide on the Glycan array. And he could quantify this affinity change to a threefold decrease in affinity. For the alpha-3 sialyl LACNAC, this trisaccharide, it could clearly obtain a 10-fold increase in binding affinity in the presence of lambda-5 unique region. So there is an increase in there is a change in binding affinity induced by lambda-5 UR, and we wanted to know the structural basis for Galectin-1 decreased affinity for LNNT. So we recorded specific NMR experiments, the HSQC experiment shown here. Basically, for those who are not familiar with this experiment, each peak of this spectrum represents an amino acid. The position of this peak on the spectrum is dependent on its environment. So if a partner is interacting with the protein and this residue is involved in the interaction, then the position of the peak is going to shift. And it is exactly what we see here. When we add to Galectin-1 the LNNT, we see a clear chemical shift perturbation on the NMR spectrum. And when we map this chemical shift perturbation on the Galectin-1 structure, we can see that it clusters within the carbohydrate binding site, which was quite expected. But when we do exactly the same experiment, but in the presence of the lambda-5 UR peptide, then we see very weak chemical shift perturbation. And if we map this weak perturbation on the structure of Galectin-1, we can see that all the perturbation observed here completely vanished. So this experiment clearly showed that binding of the peptide induces changes within the carbohydrate binding site that impairs binding to LNNT. And in order to confirm this, we studied the LNNT using NMR. We assigned all the resonances of the tetrasaccharide, and we recorded 2D STD experiments in order to map the Galectin binding site on the saccharide. So what we see here cycled are the protons that are interacting with Galectin-1. And when we do the same experiment, but in the presence of lambda-5 unique region, then we can clearly see that the contacts with the Galectin are decreased. You have less hydrogen involved in the interaction when the peptide is present. So clearly, this experiment showed that lambda-5 unique region, by binding to Galectin-1, induces changes in the carbohydrate binding site that leads to decreased intermolecular contact with the glycan. So this finding, at this point, we were convinced that binding of this protein to Galectin-1 changes this affinity for carbohydrates. And of course, we thought, or maybe this could be a way for Galectin to target specific ligands at the cell surface. But what is seen in an NMR tube is not what exactly can occur at the cell surface. So we had to think about an experiment to be able to do NMR and working with physiological Galectin-1 cell surface ligands. And this was the work of Pauline, who was a PhD student in my lab. And she basically developed a method where she cultured pre-B or stroma cell lines. And using sonication and ultracentrifugation, she was able to get homogeneous sample of cell vesicles, so mini cells, containing proteins and also glycosylated proteins. And here you can see these vesicles pictured using negative stain electron microscopy. And when these vesicles are incubated with N15 labeled Galectin-1, we can record NMR experiment in order to monitor interaction of Galectin-1 with cell surface ligands. And this is exactly what we got. We exactly saw a chemical shift perturbation when we incubated Galectin-1 with pre-B cell vesicles, as shown here with chemical shift perturbation. So we did that with previous cell vesicles. We also recorded experiment with stromal cell vesicles, and we could clearly see chemical shift perturbation. And we plotted here on this plot for each individual residues of Galectin-1, the chemical shift perturbation observed on the NMR spectra. In green, in the presence of previous cell vesicles, and in orange with stromal cell vesicles. And in order to validate the fact that the binding seen between Galectin-1 and these vesicles are glycan dependent, we treated the cells with PNGSF and also O-glycosidase in order to remove glycans. And you can see here in blue that when we treated the cell, we cannot see significant chemical shift perturbation as compared to untreated vesicles. And in order to understand the structural basis of this increased affinity for alpha-3 cell-related glycans, we had a collaboration with Lynn Colescott from CASIA in San Diego, where they synthesized for us this pentasaccharide, the alpha-3 celluldilacnac that we call SDLN. And they synthesized this pentasaccharide as C13 galactose containing glycan. So here is the NMR spectra of the labeled SDLN. And we use this sample in order to study the interaction with Galectin-1 and the effect of lambda-5-UR binding. So first of all, we did STD experiments, STD NMR experiments, so to identify on the glycan the protons that are interacting with the galectin. And by doing so, we could see that mainly the terminal galactose is interacting with Galectin-1. And when we do the same experiment, but plus lambda-5 unique region, then we see that the second galactose is also involved in the interaction. We see that the first galactose and the second galactose interact with Galectin-1. So basically, it seems like lambda-5-UR induces a change in the carbohydrate binding site that leads to a conversion of Galectin-1 from an exotype lectin to an endotype lectin. And we recorded some other NMR experiments, HSQC experiment on this SDLN, C13 SDLN sample. We added Galectin-1 and we saw that several peaks disappeared, meaning that the residues are involved in the interaction with Galectin-1, and all those resonances belong to the first galactose. And when we look at when we do the same experiment, but by looking at what's happening on the protein, we see that the chemical shift perturbation of Galectin-1 in the presence of the SDLN mainly converge to subsite C of the carbohydrate binding site. And when we do the same experiment, but in the presence of the lambda-5 unique region, then we can see that galactose 1 is impacted, but also galactose 2 is impacted. And when we do the reverse experiment by looking at the perturbation on Galectin-1 spectra in the presence of SDLN and the peptide, we clearly see that the perturbation within the carbohydrate binding site have increased, impacting CBS subsite C, but also in CBS subsite D, and even to subsite B and A. So what we can see here is that lambda-5-UR binding to Galectin-1 induces chemical shift perturbation, therefore indicating increased intermolecular contact between Galectin-1 and the glycan. So this finding led us to rethink the previous sinus model. And basically the first step of this model is the secretion of Galectin-1 by the stromal cell, which is going to interact with glycosylated receptor on stromal and privy cells, mediating cell-cell recognition. And then when the Galectin-1 interact with the privy cell receptor, the interaction of Galectin-1 with the glycosylated receptor changes towards receptors presenting alpha-3 cellular glycans. And this helps the clustering of the privy CR and induces cell signaling. And what we think also is that the concentration of specific receptors at a local, in a local environment on the membrane induces mechanical stress. And this mechanical stress can induce membrane curvature. And this could lead to pre-BCR internalization that is usually observed after pre-BCR activation, but also to pre-B synapse dissociation in order for the pre-B cell to proceed to the next step of the differentiation and associating to different type of stromal cell. So what we see here with our data is that galectin interacting with non-glycosylated proteins can drive the target specificity of galectins at the cell surfaces. But in the case of the privy cell receptor, we were really convinced actually, but we thought since galectin-1 is involved in so many different processes, does it also occurs in another process? So for that, we collaborated with Victor Tichsen in Amsterdam University, and Victor actually found that galectin-1 can interact with a chemokine, CXCL4, and we wondered if what we see with the privy cell receptor is also valid for CXCL4 binding to galectin-1. So we did NMR experiments, and we could clearly see chemical shift perturbations upon binding of CXCL4. We identified residue of galectin-1 interacting with CXCL4, and we were even able to generate NMR-based structure between galectin-1 and CXCL4. And the structure obtained using NMR data show that CXCL4 binds to the edge of the convex side of the CRD of galectin-1, which is more or less the same interacting surface than the privy cell receptor interacting surface. And as in the case of the privy cell receptor, the carbohydrate binding site is distinct from the CXCL4 binding site on galectin-1. So what then Victor did with his collaborators is to check if binding of CXCL4 can change binding to the binding properties of galectin-1 to glycans. And for that, they used fluorescent anisotropy using fluorescent saccharide probe. And they could see that in the presence of CXCL4, there is an increase, a tenfold increase in affinity for this high affinity probe. And for this low affinity probe, you could see that with galectin-1 is not able to bind to this probe, but in the presence of CXCL4, they could clearly see an increase in interaction to this LNMC probe. And in terms of function, they also tested the pro-apoptotic activity of galectin-1 on Jurkat T cells, and they could see that CXCL4 potentiate the apoptotic activity of galectin-1 on those cells. And they could also see that it is glycan dependent, because in the presence of lactose, they could not see any change. And they also see increase in apoptotic activity using PBMCs. And what was also interesting, according to their data, is that, okay, Galectin-1 binding to CXCL4 show increased pro-apoptotic activity on CD3 plus cells, but looking at subset of subpopulation within the CD3 plus cells, it could see that the effect is only present on a subset of cells, the CD3 plus CD8 plus cells, whereas in CD3 plus CD4 plus cells, it's not the case. So basically, what we can see here is that protein binding to Galectin-1 can modulate activity of galectins at the cellular level. So I hope at the end of this presentation that I convinced you that Galectin-1, this interacting site that we highlighted with protein, is really a platform to help glycan decoding function of galectin. And also this new interacting surface can be also a way to think differently the development of specific galectin inhibitors. And well, I think we will have in the next years new example of the fine-tuning of galectin activities through this interacting platform. And I would like to finish, I don't know, I would like to finish with by acknowledging people involved in the work, all the people from my lab, Françoise, Jérémy, Pauline, and Bastien, who worked quite a lot in optimizing all the NMR experiment and also the enzymatic treatment of the cells, and also the other members for discussion, idea, and support. Our long-term collaborators from Claudine, Stéphane Mancini for all the cellular biology, Stéphane Betsy for the ITC experiments, Lara for the lectin microarray, and Lincoln Scott for glycan synthesis. And of course, because as you could see, NMR is quite central to our studies, the NMR equipment managed by Olivier Bornet was very essential to the success of this project. And thank you for your attention.